Hello, this is Gina Piscatelli with the third lecture for Week 6 in Anatomy and Physiology 2 at Madison Area Technical College. In this lecture, we'll talk about how pulmonary ventilation is controlled. And this video that's showing a cadaver photograph, there is an instructor who is pointing out the phrenic nerve that travels kind of behind the heart and medial to the lungs, medial and posterior to the lungs. So she's moving away the left lung so that you can see the phrenic nerve continuing inferiorly to innervate the diaphragm. And the diaphragm, as you know, is the principal respiratory muscle. So in this lecture, we're going to talk about the anatomy of neural pathways that control those muscles, and then we're going to talk about how they function in automatic involuntary breathing. So the nervous system controls respiration by controlling those respiratory muscles. And interestingly, even though we breathe without thinking, the respiratory muscles are composed of skeletal muscle. So therefore, the neurons that are sending signals to these muscles are considered somatic motor neurons. And they travel, instead of autonomic, and they travel in spinal nerves from the cervical spinal cord to the diaphragm, and from the thoracic spinal cord and spinal nerves to the intercostal muscles. Now, the nerve that carries information to the diaphragm or innervates the diaphragm is called the phrenic nerve. And in this picture, you can see it emanating. It's got a blue arrow pointing to it, and it's emanating from the cervical spinal cord. It actually consists of neurons that emerge in C3, C4, and C5 spinal nerves. If you recall from A and P1, those spinal nerves are part of the brachial plexus. So the brachial plexus has a nerve that comes off of it called the phrenic nerve that travels posterior to the sternum and then to the diaphragm. And you can see the blue arrow on the bottom where the yellow neuron is innervating the diaphragm. And the intercostal nerves that innervate the external intercostal muscles emanate or emerge from the thoracic spinal cord, T1 through T12. Although this picture shows T1 through T7, there are actually spinal nerves all the way to thoracic spinal nerve 12 that innervate breathing muscles. Now, we can consciously control respiration, especially since these muscles are skeletal muscles and those are somatic motor neurons. I think you would suspect that, and you're human, so you know you can control how fast and how often you breathe. The way we do that is that our cerebrum in the primary motor cortex has primary motor neurons that travel down to the medulla oblongata and then past the medulla oblongata out the spinal cord. So we know that we can control it consciously, but we're going to really focus mostly on unconscious control of respiration in this lecture. So in unconscious control of respiration, let's first look at the neural pathways that are motor pathways to those respiratory muscles. The signals come from respiratory centers that are in the brain, in the medulla oblongata and the pons. So higher brain centers, of course, can influence these respiratory centers, like the hypothalamus when we're in pain, or when we get hot, we might increase our breathing, or when we get angry, we might increase our breathing rate. But the respiratory center can function without the higher brain center, and that's primarily in the medulla oblongata, and you can see two orange nuclei there that control breathing without our conscious thought. There's also a respiratory center in the pons, a little bit higher up, that basically modulates the medulla oblongata's respiratory center. Now, our breathing rate can change without our conscious input. So there must be some sensory signals sent to this respiratory center in the medulla oblongata. Things like, what's the oxygenation level of our blood? Is there some sort of airway obstruction? Do we need to cough? Do we need to sneeze? So let's now look at unconscious control of respiration, but the sensory signals that send information up to these respiratory centers in the medulla oblongata. All the sensory signals are carried in cranial nerves. Cranial nerve 9, which is the glossopharyngeal nerve, and cranial nerve 10, which is the vagus nerve, on both sides, so bilaterally, vagus nerves and glossopharyngeal nerves. Now they're carrying signals from specific receptors located in the lungs, in the airway, but also in these vessels that are shown, the aorta and the carotid arteries that are shown here. There are some special receptors called the carotid body and the aortic bodies that can detect blood levels of CO2, pH, and oxygen. And we'll talk about that more. But for now, I wanted you to know that any sensory information, whether it's coming from these carotid bodies or aortic bodies, any sensory information from lung and airway is going to travel in one of these two cranial nerves. Okay, let's examine the respiratory center in the medulla oblongata and the pons in a little bit more detail. The medulla oblongata actually has these two neuron groupings that we call nuclei. One is called the ventral respiratory group, often abbreviated VRG, because it's ventral, which is this way. That's ventral or anterior. And you can see the cerebellum peeking here, so you know that's posterior to the right, or dorsal. So one of them, the medulla nuclei, is the VRG. The other one is the dorsal respiratory group, because of course it is more dorsal. Now they have different functions. It is the VRG that is most important in establishing the rhythm that's unconscious and innate to our brainstem, so we can breathe without thinking. And so this VRG, excuse me, establishes our normal breathing rate with quiet, undisturbed respiration of 12 to 16 breaths per minute. But the DRG is the one that receives sensory information from the blood, from the lungs, even from the cerebral spinal fluid and the airways. So what that means is that this DRG can send signals, either excitatory or inhibitory, to the VRG to make it change its rhythm. Now the pons has another group of neurons called the pontine respiratory group, right here. And it modulates the transitions between inspiration and expiration. So how long it takes you to inspire, or is there a gap between inspiration and expiration where neither is happening? So that's what the pons is for. And this is particularly important in sleep, exercise, and speaking or vocalization. So if we look at the VRG in a little bit more detail, and I have it circled in red here, we know that there are two types of neurons there. One type is called an inspiratory neuron. The other type is called an expiratory neuron. It's the inspiratory neuron that really controls your breathing rate because it is excitatory to your inspiratory muscles, the diaphragm and the external intercostal muscles. What the expiratory neurons do is just inhibit them. So the expiratory neurons would inhibit an inspiratory neuron, and therefore the muscles wouldn't be excited. Now, the way that these neurons work in tandem is the inspiratory neurons generally fire excitatory impulses to the muscles for about 2 seconds, and then the expiratory neurons inhibit the inspiratory neurons for about 3 seconds. So there's one that we really don't know exactly how they work, but there's one hypothesis that states both sets of neurons are somewhat autorhythmic, a lot like the pacemaker cells of the heart, although they haven't actually found channels that are open and spontaneous depolarization due to an ion channel, but that's one hypothesis. Now let's look at the DRG, or the dorsal respiratory group, and I have that one circled in red here. We already know that it sends signals to modulate the ventral respiratory group so that whatever is sent out from the ventral respiratory group is adequate for what state the body is in. So the DRG has to receive sensory input, and it does so from the respiratory tract, from the lungs, and from the bloodstream, as well as the cerebral spinal fluid. And just to make sure that you understand about the pons and its role as a respiratory center, I'm going to repeat what the pontine respiratory group does. It modulates the transition between the inspiratory and expiratory phases of ventilation. And as I mentioned before, this is kind of important in sleep, exercise, and speaking, but it also happens with emotions. Sometimes you increase your rate when you get anxious. That's one particular example. Another important example is that when we sleep, we go through different sleep cycles throughout the night. One is called REM sleep, one cycle of rapid eye movement sleep, and the other is non-REM, or non-rapid eye movement. In one you dream, in one you don't. I don't know a lot about sleep, but that's the general idea. And it's the pons that can modulate the transition between REM and non-REM. So obviously, in one or the other, we must have to breathe more frequently. I can't tell you which one it is, though. So the pons just acts to modulate the transition between inspiration and expiration. Okay, let's look at the sensory information that is sent to that dorsal respiratory group. Some of the sensory information could be categorized as like a physical thing, a physical factor. Other sensory information might be a chemical of some sort. And then, of course, we have anything from the higher brain centers that could be sent to the dorsal respiratory group, including emotions like anxiety, anger, rest, peacefulness, whatever, pain, body temperature, and then we can also voluntarily control our respiration from the cerebrum. So we're going to mainly look at the physical factors and the chemical factors. So the two physical factors that we will look at will be the degree of stretch primarily in the lungs and the pleural membranes, and irritants that might exist in the lungs or in the airway. These might be physical objects like mucus, or I guess it could be something a little bit more serious like a bacteria, or you've inhaled something, like dust or whatever. Then there are chemical factors, and these chemical factors are innate to our homeostasis. We need to maintain a certain level of CO2, oxygen, and hydrogen ions, or pH, in our tissues and our bloodstream and our cerebral spinal fluid and all the fluids in our bodies actually. So these three chemicals are the ones that are going to be important and might trigger sensory information being sent to the dorsal respiratory group. So let's first look at the physical factors that influence respiration, or the rate of ventilation. The inflation reflex is particularly important because it deals with stretch, and its purpose is to prevent overinflation of the lungs. So there are stretch receptors located in the airways, as well as the pleura surrounding the lungs. And if they are stimulated by being stretched too much, they send an inhibitory signal to the dorsal respiratory group. And the function, of course, would be to inhibit respiration in order to prevent overinflation. Now, there's also an irritant reflex that's very similar. There are receptors again in the airway and in the alveoli, as well as the pleura, that can detect mucus, debris, noxious fumes, which I guess would actually be sort of a chemical. And a signal is again sent by the vagus nerve to the DRG. And here's where things get a little bit more complicated, because what will actually happen, the action, is that the bronchioles will constrict and there will be stimulation of cough or sneeze. So that means the diaphragm has to contract. So although the vagus nerve is going to send the sensory information to the DRG in the medulla, there is going to be a motor reflex that goes to the bronchioles, smooth muscle. So that will be an autonomic neuron to the smooth muscle, because again, in bronchioles, for them to constrict, the smooth muscle has to contract. But the cough or the sneeze will be somatic motor, of course, just like before with respiration. It'll just be very forceful. Now, the chemical factors that can influence our ventilation include, as I mentioned, CO2, oxygen, and pH. So let's look at the different stimuli and talk about these stimulants a little bit. The most powerful stimulant in controlling respiration has to do with the level of carbon dioxide, not oxygen, but carbon dioxide in arterial blood and cerebral spinal fluid. Those two fluids. And interestingly enough, CO2 causes a drop in pH, or it causes the blood or cerebral spinal fluid to become acidic. And so I'm going to ask you to remember this particular equation so that you understand the link between carbon dioxide and pH. Carbon dioxide joins with water in the blood or in the cerebral spinal fluid to form this acid, and that acid is called carbonic acid. And that carbonic acid can spontaneously dissociate into two ions, hydrogen ions and bicarbonate ions. And of course, it's the hydrogen ion that will change the pH. So CO2 is closely regulated, I guess, for two reasons. One is just in and of itself, CO2 is a waste product. The other is because it causes acidic conditions. The other stimulus is levels of oxygen, or PO2, and that is usually in the arterial blood. But it really only becomes a stimulant when it gets super low, below 60 millimeters of mercury, which is quite low. So the receptors, as I mentioned, that can detect the levels of CO2, hydrogen ions, and oxygen are located in the aortic and carotid arteries, or the aorta and the carotid arteries, and they're called aortic bodies and carotid bodies. These are technically peripheral chemoreceptors. They're out in the periphery of the body versus in the brain. There's also a set of receptors in the medulla oblongata that can detect these stimuli in cerebral spinal fluid. So cerebral spinal fluid surrounds the brain, and if the CSF has too much CO2, these central chemoreceptors in the medulla oblongata will be stimulated. They will bind and send a signal to our breathing center. So let's look at the central chemoreceptor first. I suppose it's the hardest to put your head around, kind of. In this picture, the brain is like beige. That's the medulla oblongata. And the blue is where the cerebral spinal fluid is. So the subarachnoid space, or deeper in the ventricle. And remember, in the ventricle, there's a choroid plexus that is carrying blood inside the ventricle. And then there's also arteries surrounding the outside of the brain that dive deep into the brain. So this is the bloodstream. All right. So it's not really the bloodstream that these medulla oblongata chemoreceptors monitor. It's the cerebral spinal fluid in between. So this cerebral spinal fluid is formed from the choroid plexus, and so its components will be similar to what's in the blood, arterial blood anyway, in the brain. And what these central chemoreceptors will bind to and detect and be stimulated by include a rise in carbon dioxide or a decrease in pH. And what's going to happen is the breathing rate will be increased. And that is all in with the purpose in mind to expel CO2. The peripheral chemoreceptors work similarly, except I talked about the fact that since they're in the periphery and not close to the brain, the signals are going to have to be sent to the brain by these cranial nerves, cranial nerve 9 and 10. And these peripheral chemoreceptors are located up in the internal carotid artery and the aortic body. And they're cells that detect levels of hydrogen ions, carbon dioxide, and oxygen. And they will be stimulated when the pH gets low, carbon dioxide gets high, or oxygen gets too low. And again, that's below 60 millimeters of mercury. And so a signal will be sent to the brain and the medulla oblongata respiratory center and cause an increase in breathing rate. So I thought I'd just summarize that a little bit for you, because that's probably the most complicated part of this lecture. And that is, what are the chemical stimuli that monitor or modulate and affect our breathing rate, respiration? It's CO2 and hydrogen ions in the cerebral spinal fluid that really causes the greatest degree of regulation. So this is the most tightly controlled fluid, and the central chemoreceptors are right there. It doesn't take very long either for them to detect and send a signal to the medulla oblongata to change respiration. And secondly, levels of CO2, hydrogen, and oxygen are monitored in the arterial blood by those peripheral chemoreceptors in arteries. That's the end of your third lecture for week 6, and thank you very much for listening.